with Max Kermel, where he did some really lovely microscopy, mainly investigating immunology of lung metastasis that he'll in his seminar. Since 2018, he's been an assistant professor at Fred Hutch with affiliation at UW. And he's won several awards already, including Mediviver Early Career Investigator, Hartwell Investigator, and Kavli Fellow from the Academy of Sciences. So in his current research program, he's already done some phenomenal work in terms of lung tumor biology, but also in pioneering an important technique called infinity flow, which we heard actually in a seminar last week about, where he was using with a large international team to develop this machine learning combined with highly multi-parameter flow cytometry to do really large scale immune profiling. And what I've admired about Mark's science consistently is this sort of innovative approaches with really sort of deep thinking. My first encounter with Mark was on a Google Doc where we were co-authoring a perspective piece. And I noticed how thoughtful he was sort of about the practice of science as well as the science he was doing. And then he generously gave me lots of advice on dendritic cell phenotyping for a recent publication. So yeah, I'm just thrilled that he's here, and I hope you'll all enjoy his seminar and ask him lots of questions.
Okay, thank you so very much. This is such an incredible honor to be here. I have never been to UMN, but it's an institution that I've been extremely familiar with as an immunologist for many years, and such a pleasure to come and share what we've been doing. So yeah, as Tanya said, and thank you for the very kind introduction, I am going to share some of the work that we've been doing over the past several years to try to understand sort of the intricacies of immune regulation of pulmonary metastasis. I will say up front, I am very much at heart an immunologist, so you may notice a theme as we go through that I tend to come back to maybe some more fundamental immunology questions as I'm trying to think about how these cells behave and regulate in pulmonary metastasis. And the other thing I'll say to preface is I'll share a little bit of my older work that's published at the beginning, but pretty much everything else is going to be unpublished. It's a story we've been working on for a while and is kind of coming to the point where I think we're ready to publish it. But as such, you know, as we get towards the tail end, there's a lot of stuff that we're still exploring and questions that we are kind of delving into that we don't have all the answers. So please feel free to raise your hand or just yell at me as I'm talking and ask questions. I love feedback and input as we go, or we can save it till the end, as you like. Okay, so to start with lung metastasis, I just kind of want to set the stage for why one of the main arenas that I think about immunology in is this process of metastasis rather than cancer in general. So why study metastasis? And there is a fundamental reason that it's actually for most cancers, not all cancers. It's the case that the primary cancer is rarely the cause of death in patients. And even in some cases, the majority of the morbidity is not due to the primary cancer. Rather, as that cancer spreads to other sites in the body, like the bone, the lung, the liver, the brain, is when you start to see the most impact in patient populations, and the cancer metastasis is the leading cause of death in cancer. The other facet that I find personally very interesting, historically we often group these things together, but the reality is that from an immunologic perspective, as well as a cancer biology perspective, the primary tumor and the microenvironment of that tumor really can be quite distinct from that of the metastatic site. The features that cells have to go through and adapt to survive in circulation, for example, rather than in the primary site where they've sort of evolved, are quite different, and they lead to different immunologic consequences that I personally find very interesting. So why study lung metastasis in general? The true answer for this for me personally is I've spent my whole career studying the lung, and I love the lung, and I continue to not understand much of the immunology of the lung because it's so diverse and situational. But fortuitously for, the grant funding writing that I do, nearly all major cancers can and do metastasize to the lung. It turns out to be a particularly sensitive site for cancer metastasis for reasons we can get into. I won't really dabble on that. It's not something we entirely understand, but there are features of that we do understand. So it ends up being a particularly relevant site for both the immune system and cancer convergence. And lastly, it really does have a significant impact on patients. So I wouldn't go so far as to say that metastasis to the lung is the worst possible outcome in cancer metastasis, but it certainly is incredibly harmful. And I would venture to guess that many people in this room and on the audience have really huge health issue in our world that we need to know more and more about.
You can actually get rid of that little thing that's blocking your title. No, I don't know how to do that. Okay.
It's not hearing anything. Turn up volume. Maybe if I turn up the volume.
I can take off your pointer.
Sorry to interrupt. Do you know how to do that? Apologies for the interruption.
Totally fine. I just invited interruptions. That's where... When I go over time now, I can blame something. Okay, well, I'll partly keep talking while we're solving that.
Yep.
Okay.
It's just local.
It's just local. Okay. I will try to read out the titles if they're relevant, and I'm not going to say them in general. So that just says lung metastasis of cancer up there. I think the online audience is probably not seeing it. Nope, but now my slides are not advancing. Hello, there we go. Okay, so this is about the amount of context I'm going to give you on what we call the metastatic tumor microenvironment. So much of my work since I was a postdoc and into my current lab has been focused on understanding this entity that is called the tumor microenvironment, particularly the immune tumor microenvironment. And the way I think of this is cancer cells don't succeed in isolation. So, we think of these cells as, these little pathogenic entities that grow in our body. In the process of metastasis, they escape from the primary tumor, they enter the circulation, travel to distal sites like the lung, set up shop, grow, proliferate, damage, cause all sorts of downstream consequences. The reality is that without the cells that coexist in either of those sites, in the primary tumor, in circulation, and in the metastatic site, those cells likely wouldn't succeed. They need these kind of support signals from the local stroma, from immune cells. They're also actively evading the immune system in particular. Our bodies are actually, turns out, we've learned quite efficient at detecting and eradicating these cells. And so the successful tumor cells have developed mechanisms to promote tolerance, essentially. So one of the things that we've tried to do since I entered this field is understand a variety of questions, in particular, kind of picking apart the different facets of the immune microenvironment of the tumor and trying to understand sort of the individual contributions of immune subsets, not just in isolation, but how these cells interact with each other in that environment and perpetuate specific responses. The other major question is how does the immune system react to the tumor? So how does the environment of the tumor coax out different phenotypes and different functions from the immune cells? And how does that in turn feed into both mechanisms of metastatic support and anti-metastatic systems? And lastly, and what I'm going to talk about a bit today, or at least this is the context in which the project I'm going to speak on, is if we do have these potent anti-metastatic immune responses, why in a patient that has metastasis are those failing to protect the patient? I don't have the answer to that, just to be clear, but that's kind of the context in which we're thinking. So generally speaking, some of the work we've previously done sort of classifies certain axes of immune cells is either sort of pro-metastatic or anti-metastatic. On the pro-metastatic side, you have a monocyte macrophage axis, and I will be very careful in saying, one, this is a bit of an old slide, but actually I believe this axis can be converted to being an anti-metastatic axis as well, and very likely there are both pro and anti-metastatic features of monocytes and macrophages that are important. Certainly, patrolling monocytes, which are close relatives of the more common conventional monocytes we think about, have been shown to have potent anti-metastatic properties. And then the major focus of what I'll talk about today is dendritic cells, which we think similarly can have kind of a dichotomous role. Okay. So to speak about some of what we did in the past, one of the tools that we developed and utilized to start understanding sort of the dynamic immune tumor interactions early in vivo in the process of metastasis was intravital microscopy. This is very much for those who are familiar with my postdoctoral mentor, Max Krummel. This is sort of his neck of the woods and something he has spent a great deal of time and effort developing, and as an excellent educator for a postdoc in learning these processes. So in my time in Max's lab, I helped develop a system where we're able to do intravital lung microscopy. That was just a general schematic. I'll show you very briefly here the approach. So this is a surgical approach where we implant a vacuum stabilization window between two ribs over the lung. So you can see we make an intercostal incision. We have sort of a flanged attachment here that slides between the two ribs, sort of like a rib spreader. We can apply very gentle vacuum pressure here, which stabilizes the lung. So the reason we need to do that, as you might imagine, if we want to image a living organism and the lungs of that living organism, respiration still needs to occur. So we don't want to stop respiration. So these mice are on anesthesia. We have them on a ventilator so that they're continuing to respire relatively normally. And we've somewhat immobilized a small region of the lung. And I say somewhat because actually we do allow inhalation and exhalation and normal respiration to occur. It's just doing it on a finer scale so we can actually image it. I'm not going to show too much of our data that we've collected with that, but I want to show a couple of key examples. So one of the major features that we noted as we were kind of probing the very earliest hours of metastasis was that the immune system is very much in a kind of seek and explore kind of role, where in this movie I'm going to show you here, I'll just hit play, assuming it will play. Yep. Maybe not. I might need to get to the pointer, which I'm not seemingly able to do. Yeah. I tried space and that's advancing. This is usually why I use my own laptop, but it failed me today. Oh, here we go, there we go. Do you know what's going on there? Sorry, everybody, I apologize for this. If not, I'll just describe it and we just won't have the cool, fun movie. Well, so essentially what happens in this movie, and we'll see if we can get it up. Oh, there we go. There we go. Okay, awesome. Thank you. Let me get rid of these two slugs up here. Go ahead and talk. Okay. So this is a tumor cell that's been in the lung. This is a B16F10 melanoma cell that we've expressed the fluorescent marker ZS green, which is a fluorescent protein that's very stable, very bright, useful for all sorts of purposes. So, nope. Sorry. Nope, you're fine. Okay. Go back. Okay. So this cell has been in the lung for about 4 hours at this point, and we're starting to watch the very earliest moments where monocytes are sensing the localization of this cell, the fact that it's arrested in a blood vessel in the lung. I'm not going to talk about this, but we know these cells make lots of microparticles which influence the immune response. And you can see one of these microparticles at the tail end gets targeted and kind of swarmed by these monocytes. So this led us down a set of questions where we started to ask that first question I showed about how does encounter with the tumor actually influence the immune cells in that tissue. I'm not going to talk too much about that today. We have a couple of publications showing that actually macrophages in particular that ingest tumor material are altered by this process. And this is similar to work that Miriam Murad has published recently, that actually that process of ingestion and sensing of microparticles or apoptotic bodies has a appreciable effect on the phenotype and function of the immune cells. What I will show here to kind of lead into my next part of this talk is that particular fluorescent protein ZS green that I mentioned. I feel like I'm just losing pointers left and right. There we go. Not showing up. Yeah. Has a very nice characteristic for the purposes of tracking immune ingestion. So it's an incredibly stable, both from a perspective of protease resistance as well as pH sensitivity. So it can go into a macrophage lysosome and it can stay bright green fluorescent. So we can actually track these cells for days, which is not the case with, say, GFP. Other cells like dendritic cells, which I'll talk about soon, you can do a pretty good job with them, Cherry and GFP. The nice thing about ZS green in that context is it's exceptionally bright, so we can get a nice delta. Using this and flow cytometry, we can actually map the cells over time that are taking up tumor material. There's various ways we can interpret that. The way I usually phrase it is we know these cells have engaged with a tumor cell in some capacity. We don't necessarily know that they're still actively localized next to a live tumor cell, for example, but we know that they've had an encounter with tumor. So what we found when we mapped sort of over the course of the first 24 hours of a metastatic process was this sort of wave-like effect where you see an early recruitment of neutrophils. This is almost certainly because of the model we're using. I don't want to overblow the fact that we get a huge influx of neutrophils. We have replicated some of this work in more spontaneous models, but what I'm showing you here is an IV metastasis model where we're doing a bolus implantation. That's going to spur a neutrophil responses. There's no way around it. But then subsequent to that, several hours later, we start to see the first wave of recruitment of conventional monocytes. Again, in earlier versions of how we thought about this was largely a pro-metastatic axis. We know that this is active recruitment. It's driven by the chemokine receptor ligand pair CCR2 and CCL2 largely. Those cells come in, and you'll note that by between 4 and 6 hours, they've kind of waned off, and that's not because the majority of them have left. It's because they've transitioned to something else. I'm going to skip over petroleum monocytes for the moment, and that transition is actually into macrophages. So these monocyte conventional monocytes are actively engaged in a differentiation process, we believe influenced by the tumor, to become macrophages that, again, we largely think support the survival of those tumor cells. In a project we're working on right now that I'm not going to talk about today, we've actually performed a very detailed single-cell RNA-seq kinetic using fixed cells to try to actually map in a very fine detail this process of differentiation. And what's been really striking to us in that process is, which is very much still in the middle of, is how rapidly this transition happens. So we often think of this as a multi-day process, monocytes going to macrophage. In our hands, at least, it happens on the order of hours. In between 4 and 8 hours after recruitment, we see things that look largely like mature macrophages, but we think they continue to evolve over time. So the important bit here for what I will share for the rest of the talk is kind of one of the big surprises for me personally in this data was that we started to see quite early as 2 hours, I'll show you in a moment, loading of dendritic cells with tumor material. And I'll go into why that was a surprise a little bit later, but this became a kind of a central focus for me, is wondering, okay, what role are dendritic cells playing a role in the metastatic process, particularly early on, and what role might that be? I'll come back to describing a little bit more about what dendritic cells are in a moment, just for those who maybe aren't familiar. So this is the data I just hinted at. So if we track tumor ingestion using the ZS green fluorophore over the first 24 hours, you can see an appreciable amount of ZS green as early as 2 hours. It kind of peaks out around 6. We've actually mapped this in more detail now. This is a bit of an older figure, but it really peaks around 12 to 16 hours. And then kind of recedes by about 24. If we use that same intravital microscopy approach that I've already described, this is looking at 24 hours. DCs are labeled in red, tumor cell in green again. You can see that these cells are actually directly interacting at some level with the tumor cells, and hopefully you can appreciate these small little blebs of tumor material that have actually entered into the DC. This is a process of direct sampling, and we've looked at this various ways at this point. Importantly, so I will preface by saying I would say at this point that this system that we used back in 2016 is a bit outdated now, the ZBTB46 DTR system. Still very useful. So this was a tool developed by the Nussensweig lab to ablate dendritic cells. We've since learned that there are likely other populations of immune cells that are influenced, including ILC3s, surprisingly, but also perhaps some monocytes. So take that as it were, though I will say, in subsequent experiments with somewhat more precise tools, we've gotten similar results. So when we ablate dendritic cells early on in the process of metastasis, we've done this both in IV implantation models as well as spontaneous models. We see this truly tremendous increase in metastatic burden. So each of these black dots on these mouse lungs is a burgeoning metastasis. This is about 2 weeks after implantation. And it's not hard to appreciate how catastrophic a lack of dendritic cells is. Importantly, ZBTB46 ablates more than one kind of dendritic cell, right? So that needs to be contextualized. So circling back around to DCs, generally in the lung, we think of DCs this way in my group. There are other ways of thinking about them, but I usually subscribe to the classification of what's called a classic dendritic cell or a conventional dendritic cell, a CDC, as cells that derive from a pre-dendritic cell precursor from the bone marrow. So those CDCs can be roughly split into two subsets, CDC1s and CDC2s. Mostly, well, entirely today, I'll be talking about CDC1s. These are famously the dendritic cells that are capable of cross-priming CD8 T cell responses, so they can take exogenous material and present that on MHC class 1 to talk to CD8 T cells and activate anti-tumoral responses, antiviral responses, et cetera. We usually use a marker XCR1 to define these cells. It's fairly robust in most contexts. CDC2s are, in my personal opinion, incredibly fascinating, but a lot more diverse in their both description and phenotyping. They're a little harder to pin down. And we haven't done that as much with them in this context, but it's definitely something we've been thinking about. I say all that just to say, I think CDC2s are likely also playing a role, but we haven't delved deeply into that process. From a functional perspective, DCs, CDC1s in particular, have been described. These are the cells that initially sense viral assault on the lung, for example. As I'll show you, we believe they're the cells that initially sense tumor assault on the lung. They're capable of both presenting antigen to T cells. They can produce cytokines like IL-12 that are known to be important for activating both T cell responses and NK cell responses, and a really key element of protection. The other bit of important background here is in the primary tumor microenvironment, there's now a great deal of really excellent literature showing the important role of CDC1s within the tumor microenvironment and within the lymph nodes that are draining that site for not only the initial response, anti-tumor response endogenously, but the production of tumor-specific T cell responses that then can be reactivated with, say, immune checkpoint blockade. So these are ever growing to be a critical facet of how we can leverage the immune system to defend against cancer. At the time that we were initially doing this work, this was an unexplored facet of metastatic biology. So linking into why one of the reasons we focused on CDC1. So this is not our work. This is work from Mark Smith's group using the BATF3 deficient mice. So these mice, among other defects, but in particular, they have a profound lack of CDC1s in most contexts. For those in the know, in certain infections, you could actually get compensation and CDC1s will develop and activate. But in our hands, we've used the same tool, and in Mark's hands, this is quite a nice deficiency in CDC1s. So they get a very similar phenotype just with the lack of CDC1s in this context. I'm not going to walk through the data, but there's a survival effect. There's similar metastatic burden increases. And importantly, one thing they showed in their study is that the CDC1 mediated suppression of tumor metastasis was driven by IL-12 production from those CDC1s and NK cell activation. Those NK cells then being cytotoxic to the tumor cells that are attempting to seed the lung. I'll circle back to that in a little bit. Okay, so what I've shown you so far is basically we have a critical role for CDC1s in suppressing metastasis from an endogenous perspective. data that I didn't go into deeply, but we believe that that's largely mediated by CDC1. So there's certainly room for CDC2s to be participating in that process. So why was this striking to me as an immunologist thinking about the lung? And largely that comes down to this. So if you delve back into the literature or you just look yourself with various tools, that's not going to show up super well. Maybe this one will be better. Yeah, we'll just look at the zoom in. Quite famously in the lung, CDC1s as well as CDC2s are dominantly localized around the airways. So you see the preponderance of those cells actually around the large airways, which is interesting in and of itself. So you can see those clusters here. What's shown in this background picture more broadly and hopefully is appreciable here. So here I've used multiplex immunofluorescence to map XCR1 positive cells within a lung slice. We see this pretty much, this is a steady state lung, no tumor involved. So hopefully what you can appreciate is indeed, these are the airways. There's quite a large number of CDC1s that are localized to that site, but not entirely, right? We have these distal cells around the superficial pleural vasculature, as well as kind of in the parenchyma. And this is an important thing that doesn't reflect well in some of the schematics I'm going to show you. The lung is obviously intimately intertwined between air spaces in the alveoli and vasculature. So some of what I would show you, I'd like you to keep that in mind, that some of the patterns we see when I start talking about the localization and function of the DCs is really striking because of how intimate vascular and airway is in the lung. But regardless, this was the picture I had in my mind going into this, is that, oh wait, but how are DCs sensing metastatic insults so early when they have this dominantly airway localized phenotype? And in fact, if you then try to marry this to metastasis and ask, okay, well, if we look very early in the metastatic process, so in red here are our tumor cells, in YFP, kind of green color here are the CDC1s. Obviously, I've cherry picked this image, right? But I'll show you some other ones later that show interaction. I showed you one earlier. But the truth is, the majority of tumor cells are nowhere close to DCs. There's really a disadvantage. So this comes back to one of those questions I posed earlier, is why do we have anti-metastatic systems that perhaps are not quite efficient enough to block metastasis? And I think this is part of the equation, is perhaps the cells that need to be sensed are not proximal to the sensors. Though not entirely true, as I've already showed you. Yeah, please.
In your previous schematic with the kinetics of the myeloid populations, that was showing interaction based on this fluorescent indicator. And I'm curious for the DCs how that compares to the relative actual numbers. Are like all the DCs there showing up with the indicator, or is it only a?
It's a very small percentage. So it's roughly 10 to 20% of the CDC1. Oh, yep. So the question was, if we compare the different immune cells that we see interacting with and kind of loading with tumor material early, how does the frequency of DCs loading with tumor material compare to monocytes, which are more prevalent? So we see a high percentage of the monocytes, probably like roughly 40 to 50% of those monocytes load, and an even higher percent if we look at the macrophage progeny. The DCs is more like 10 to 20%, so, which makes it very fun to try to study those cells, I will say. Okay, so circling that idea a little bit, I just wanted to frame this question a little bit. So this is how, as a graduate student, I largely thought about the lung, is that one of the really key facets of why we have lungs, right? Respiration and providing oxygen to our tissues also leads to a key vulnerability, right? Every breath that you take brings in potential for virus, allergens, bacteria, particulates, et cetera. So this is a key vulnerable site in the lungs. And as such, dendritic cells, which serve as these key sensors and surveillors of the pathogens assaulting the lung, it makes sense that you would have a preponderance of those cells around the airways, right? However, the airways are not the only vulnerable site of entry into the lung, right? So I've already shown you that in the process of metastasis, entry through the circulation is also vulnerable in a slightly different way, right? That's the process of metastasis, but you could envision this in diseases like sepsis or malaria as well, that the lung is rife with blood vessels, and it's really truly an also sensitive spot. And we truly don't know much at this point about how surveillance of the blood vessels might occur in the lung, and that's what I'll spend the rest of the talk discussing. So we posed this question. We had evidence from this very rapid loading of DCs with tumor material. This is before those tumor cells have extravasated, well before the tumor cells have extravasated. Presumably they're in the blood vessels. So we knew that something was going on. So we asked a pretty simple question. We used a co-labeling scheme where we took fluorescent antibodies against CD45.2 actually, and injected one color of antibody intravenously, one color intratracheally. This is a fairly well-established method. I suspect many of you are familiar with it, but I'm happy to discuss it further if not. Where effectively cells that are either within those compartments, so either in the blood vessel or in the airways, would label with the antibody. Importantly, as we currently understand it, dendritic cells, at least in mice, are not in the blood vessels and very rarely in the airways themselves, but they can reach through. We know this of the airways. We've actually, my old lab imaged this directly, that, and it's also been well shown in the gut, that they can reach through epithelial barriers. So the way we're going to interpret what I'm going to show you is that likely this is some mix when we see labeling in, say, the vascular compartment. It may be that those cells are somewhat in the vessel. We think that's probably somewhat the case, but they could also be perivascular and they're actually accessing that vesicular compartment. And I'll show you an example of that with microscopy too. So this is the pattern we got when we performed flow cytometry, is we got roughly 3 populations, and they were quite distinct. This is a contour plot. It looks clean, but it honestly is pretty much always this clean. We pretty much never get co-labeled cells. The DCs so this is gated on total DCs in this case. I'll show you CDC1s in a second. We get a very clean population of vascular labeled cells here, roughly 10 to 20% of the total DCs, and a very clean population of airway labeled cells. And the vast majority kind of either pick up a little bit of the airway label or don't pick up anything, is all we usually call those unlabeled. Importantly, this is a snapshot. I'm not saying that those cells might not go on to interact with the airway or a blood vessel, though we don't think that's true of the blood vessel. But at any given moment in time, they're not doing both jobs. So we can quantify this. And if we look at the identity of those DCs that are engaged in vascular airway labeled processes, it's the majority, it's the CDC1s that are in both contexts, but there are certainly a appreciable number of CDC2s. And I will say this is based on frequency within the total population of those subsets. There are more CDC2s in the lung than there are CDC1s. So there's quite a number of CDC2s doing this as well. So what's actually happening? So we can marry that labeling approach to our microscopy approaches. I'm not going to show a movie here, but this is live lung tissue. This is now not intravital imaging, it's live explant imaging, which allows us to see deeper into the lungs and have a little bit more scope of what we're looking at. So this probably won't show up super well. I'm going to zoom in here momentarily. But importantly, the vast majority of the DCs that we see, so there are these little yellow dots here, here. The red is the IV labeled cells. So mostly what you're seeing there are neutrophils and monocytes that are picking up that IV label. They localize beautifully to the vessels if you zoom in. Most of the DCs don't pick up the vascular label. It looks just like the flow I just showed you. But if we hunt around a lot, and I do mean a lot, this takes quite a lot of effort to find. I took this movie personally because I struggled to convince my postdoc that we would ever find them, so I did it myself. Here is now zoomed in on this one cell down here. And I would say over the experiments we've done with this, we've managed to find probably 20 to 30 of these cells. It's not a huge number, so they're quite rare. You can hopefully appreciate from this front on view that there's a nice kind of membrane stain. This is the CD45 IV antibody. You can see a vascular labeled blood vessel. And this is what's important. So if you look at this now in the Z dimension, you can see that that label is restricted to where the cell is interacting directly with the blood vessel. We believe it's actually within the blood vessel, though. Resolving that's a little tough in that explant model. But you can see the majority of the cell is unlabeled, sitting kind of in this perivascular niche. And as it dips down into the vessel, it picks up that vascular label. We think this is the dominant phenotype, that you have these kind of perivascular cells that are dipped in. But we do have movies that look more like they're maybe entirely sort of marginated in the vessel. So we haven't fully answered that question. It's probably a mix of both. And I'll get into what we think is happening there in a little bit. Okay, so jumping off of that data, I think a really important question that we wanted to know is, are these distinct populations, these vascular surveilling or airway surveilling, or are they somehow interrelated? I'm going to start out showing you that they're distinct, but then I'm going to argue that they're actually on a continuum of differentiation. I personally don't believe these are like, this is not a new subset of CDC1s. This is a stepping point along the way to what we conventionally think of CDC1s. However, if we use that same in vivo labeling scheme, we sort those cells and perform bulk RNA sequencing. We leveraged a very low cell input version of this that the Veterinary Research Institute has optimized, so we can do this on our very rare cell populations without having to use huge numbers of mice. We get these very distinct transcriptional profiles. So the airway and what we call the unlabeled population generally align. There are differences. The ones that airway label look considerably more activated. For those who are familiar with Mary Murad's MREG DC work, they perhaps look a little bit like those, like the cells that are directly interacting with the airways. They produce a lot of CCL17 and CCL22, quite interesting. Whereas the blood cells kind of sit, or blood interacting cells, sit off on their own and have quite a distinct transcriptional profile. I'll show you a little bit more depth. So importantly, these are all CDC1s. They have universally share the hallmark of XCR1 expression, CLEC9A expression, IRF8 expression. They all look like classic CDC1s. They have CDLMC, MSC class 2, et cetera. The blood sampling population, quite frankly, look like a cell that is much earlier on in the differentiation process. So they still express some markers of predendritic cells, and they express like, so lefty1 and CD62L, SEL tend to be kind of expressed on earlier progenitors, say in the monocyte compartment, but also the DC compartment. However, importantly, I'm not going to show all this data, but they're functional DCs. They can prime a T cell. So we are starting to think of these as probably the first stage of DC development as these cells enter the lung. And I'll show you evidence for that in a little bit. The airway cells, as I already mentioned, have a more activated phenotype as compared to the unlabeled cells. But they do share some hallmarks. So the most interesting facet of this, in my mind so far, is they have this very clear expression profile of epithelial cell interaction. So they express high levels of the molecule EpCAM, which as immunologists, we often think is a non-immune marker. It's actually all over certain cells of the immune system. It's in Langerhans cells of the skin. It's in alveolar macrophages. I'll show you, hopefully convincingly, it's in CDC1s, at least in the lung and probably the gut as well. Cadherin-1, these cells famously express CD103, which interacts with another epithelial interaction molecule, E-cadherin, CD207, which is Langerhans, so on and so forth. So one way I interpret this, I think we found something unique here with the blood surveilling cells. The airway and the unlabeled, this is the DC, this is the CDC1 that pretty much every paper you've looked at that had lung CDC1s, if you have, these are those cells. These are not, we've maybe put a little bit of an extra definition on them with the airway population, but they're those kind of classic DCs. Cool to know that you guys had somebody talking about infinity flow recently. I'm curious who that was. But I'm not going to really describe this method. It sounds like some of you would have seen it. But the next thing we did, building off of that transcriptomic profiling, is we did cell surface protein profiling. The long and short of that is this validated a lot of what we were seeing in our RNA data, that we could see very clear expression of EpCAM on a subpopulation that CDC1s that co-expressed with CD207, with CD103. The blood surveilling cells kind of sit here. They express CD62L and CD45RB, for example. But I'm not going to go into too much depth for that. Really quickly, one of the questions I most often get asked, if I don't show this, is, hey, you're saying these cells are interacting with the epithelium. Are you just kind of stripping off these adhesion molecules as you go? Like, are they really expressing EpCAM? We believe they are. So we can see the staining pattern really clearly. But we also got EpCAM flox mice from Mark Udi's group. We now, I think, are the sole source of these mice. If anybody happens to want EpCAM flox mice, I send them out to people all the time now. We cross those to the ZBT46 Cree. So EpCAM deficiency, globally is lethal, right? So we need to restrict it. ZDC CRE does a pretty good job getting it restricted to the DCs. So in the context of this system, CRE positive mice completely lack EpCAM expression, but it's maintained on the airway epithelial cells. So we're reasonably well convinced that this is cell intrinsic EpCAM expression, as supported by our protein and RNA data as well. Importantly, both populations are dependent upon BATF3. I already told you that this is in BATF3 knockouts, you lack CDC1s. These both EpCAM high and EpCAM low CDC1s are BATF3 dependent. They are CDC1s. And then lastly, showing kind of that latter bit because we often use EpCAM as a surrogate marker for airway versus vasculature surveillance. So if we perform that same assay with the in vivo labeling and we look at the EpCAM positive cells, this is where we see our airway labeled DCs. And then very cleanly, when we look at the EpCAM negatives, that's where our vascular labeled DCs sit. So this is one piece of evidence for why I think actually the vascular population, it's not that it's transitioning back and forth and maybe they sample the airways sometimes, maybe they sample the vasculature. When they have this vascular phenotype, I think they're dominantly sampling the vasculature or at least interacting with the vasculature. Okay, so what I've shown you so far is a passive label. I want to show you a little bit that these cells actively can access those compartments and take up material. So a classic way of doing this is using fluorescent beads. So we can inject these yellow-green fluorescent fluorospheres intranasally. And when we do this, the majority of them go into alveolar macrophages, which is not a surprise. But when we look at the CDC1 compartment, not exclusively, but pretty darn near exclusively, they're all in the EpCAM high population, supporting that idea that those cells are the ones that are actively engaged with the airways and surveilling through the airways. When we do the inverse experiment and inject beads intravenously, we see the opposite, that we see the majority of the cells that manage to take up those beads are the EpCAM low. I'll say in this context, it's petroleum monocytes that take up the most, so we have to collect large numbers of cells to see these, but it's been pretty robust. I won't go into depth on this, but the take home of this, so this is our bulk RNA sequencing here. This is public single cell RNA-seq data from both mouse and human. We now have our own mouse data, but it's not represented in this slide. We can see these same cell signatures in mouse very clearly and quite cleanly in human as well, though it's a little bit harder to, it's not quite as clean, I guess. It's a little messier. But we have pretty clear populations that fit this sort of airway surveilling phenotype and blood sampling signature, and the majority being this kind of unlabeled. I don't want to overblow that. I think we need to learn a lot more about the human context. We don't, for example, see EpCAM expression in the human cells. We see other adhesion receptors that are epithelial, but there's clearly differences between mouse and man there. Obviously, we're limited in how we can interrogate the functional facets of those cells in people, so we haven't gone as deep on that yet. But we do think that there's crossover to human health as well. Okay, so all well and good. Hopefully, I've convinced you that there's something interesting going on here a little bit, that there's kind of a underappreciated surveillance mechanism of the vasculature in the lungs. So the next question that I'd like to talk about quickly is, does this actually matter for metastasis? This is something we're still very much working on. It turns out it's very hard to independently ablate these cells. I'll show you, we have a tool to kind of start to get at that at the end. But we wanted to know, okay, if we're predicting that there's this very early surveillance of metastasis mediated by CDC1s, which I showed you earlier, is it actually these vascular surveilling CDC1s that we've identified that are doing that? The answer to that is yes. So when we use that same B16F10 Zia screen tumor model, implant IV and look very early, 4 hours, we see labeling of the DCs. And when we characterize which population those are, they are dominantly the EpCAM low, not exclusively, but dominantly the EpCAM low population. I'm not showing this here, but we have married that to the IV labeling approach, and it all co-aligns that those EpCAM low cells that are taking up tumor material also label with the IV label. So not only can they take up beads, but they can take up a more relevant material like tumor material. And do so very rapidly. Lastly, I hinted at this data from Mark Smith, but the mechanism by which CDC1s are thought to mediate anti-metastatic immunity through NK cells is IL-12 production. When we do a detailed time course of IL-12 reporter, this is IL-12 YFP mice, what hopefully you can appreciate is that if you look at the EpCAM low CDC ones here, so PBS versus 4, 8, 12, 24, 72 hours post implantation with tumor, we see this really clear upregulation of IL-12 that co-aligns with some of the facets of tumor cell clearance, for example, that I'm not showing here. I don't want to state that, we do also see a late increase in the EpCAM high. We're still trying to understand that. This is at a time point when tumor cells have started to escape the vessels. Maybe they're gaining access. What I actually think is probably happening there is what I'll tell you in a moment, is we think that the EpCAM low cells are further differentiating into EpCAM high cells, but we don't know that. That's one of the questions we're still grappling with. I'll show you some data with Metastasis a little bit later, very soon, because we're low on time. I wish I could say, hey, we ablated just the EpCAM low and it was beautiful. We lost all protection. We haven't figured out how to do that yet. I've been trying for 2 years to get a clean system using DTR mice and like IV DT versus airway DT. It's not, we don't have the specificity. So apologies that I leave it there, but that's where we are currently. So this is the model we've kind of come to. Some of these things are still very much hypothesized rather than proven. But I've become very convinced that there are these two different states of CDC1s in the lung, that you have kind of a dominant airway surveilling pool, a vascular surveilling pool. And we think, and certainly have evidence to support this, that these vascular surveilling EpCAM low CDC1s are among the first sensors of metastatic assault on the lung, and they're certainly capable of activating and producing IL-12 in response. And we're working on tying that thread through to NK cells and the importance of this process. I suspect in the end, we're going to learn that both DC populations are doing something. That's usually how these things go. But I at least hypothesizing that these, excuse me, are really important for that process. Okay, so in the next few minutes, and I may have to abbreviate a little bit of this, I'm going to show you the data that I think supports that this isn't like some unique population of CDC1s that exists as its own independent, pool of cells. I really do think this is a differentiation continuum with functional consequences on either side. So part of that comes from single cell RNA sequencing. Again, this is public data from Miriam Murad's group that I reanalyzed. We now have our own data on this that supports this. It's not quite ready for prime time yet. But so if we take single cell, this is from murine lungs, I'm actually using a very similar model. And use Monocle, which is now a little bit defunct, but to do trajectory analysis. So we see our signature of the blood sampling, unlabeled, and airway sampling kind of align into this trajectory where blood sits on one side, airway on the other, with the unlabeled in between. For those of you who are familiar with pseudotime analysis, take this with the grain of salt it deserves, right? In pseudotime analysis, you're usually telling it the starting point. So I said, hey, start over here, and you get this beautiful pseudotime trajectory out to this way. That said, the trajectory exists. I could start it over here and it would run the opposite way. But for reasons I've already described, this blood sampling signature co-aligns with earlier development states. So that's why we've done it this way. But it supports that there's an interrelationship between these populations at the very least. You can look at that slightly different ways, that as you go from blood through to airway, you see upregulation of things like EpCAM and CD103, downregulations of things like plaque-8 and actually BATF3. It's not that these cells don't still express BATF3, but they are higher for that differentiation mediating transcription factor early. So we hypothesized that essentially what's happening here, and we still have a lot of work to do on this, that predendritic cells are known to enter through the blood vessels into tissues, into the lung. I will say this is known. There isn't really a ton of direct evidence, but it's hard to imagine anything else happening based on the data that exists. They enter into the tissue for reasons we don't yet understand. And we do know that they respond to FLT3 ligand in the lung, and that induces differentiation into dendritic cells. So I'll show you data to support this, but we believe that this vascular surveilling state is likely sort of that earliest state of a FLT3 ligand responding DC. So it's mature DC, but very early on. And then we believe there's some process that drives them through, and that's what I'm going to show you. So one way we've gotten at testing this is ablating all the CDC1s. So we use the XCR1 DTR system, which is a really beautiful DTR system for anybody who's interested and hasn't used it. We can get very clean ablation of not just airway and vascular, but both. And then we can follow the repopulation kinetics of those over time. When we do that and we look at EPCAM high versus low status, we see very clearly, so this is PBS, this is all cells ablated, and then as early as day 3, we see the reappearance of the EPCAM low population of cells, and then we see lagging behind that, the reappearance of the EPCAM high. Similarly, if we look at vascular labeling versus airway labeling, we see roughly the same thing, that as soon as we see those CDC1s reappear, they are labeled with the vasculature. I suspect in this early phase, they're probably actually fully in the vessels and migrating out, though we don't know that for sure. And then the airway labeling capacity lags quite a bit behind, as those cells presumably are finding their way through the tissue to the airways. So to start to try to pick that apart and understand what's going on there, we went back to our RNA sequencing data. We did pathway analysis and we compared airway to blood or unlabeled and airway to blood, et cetera. We get the same result. So there's this very distinct and very clear signature for TGF-beta signaling in the airway and unlabeled cells versus the blood. I'm not going to go in depth into what TGF-beta is, just in the interest of time. But hopefully everybody's here familiar with it at some level. As immunologists, we often think of it as an immunosuppressive cytokine. As cancer biologists, maybe we think of it more as a differentiation factor, or even in developmental biology. It's both of those things. It does both of those things. Our interest here is that as a differentiation factor, though, I would never argue that the immunosuppressive function is not also critical for what I'm about to show you. So just keep that in mind. Very clear signature of TGF-beta though. So we initially modeled this in vitro. So we have pretty decent ways of growing CDC1s in culture, thanks to work from Tim Sparwasser and Miriam Murad. So we leverage these systems which use timed administration of FLT3 ligand and GM-CSF. What I'm showing you here is just phenotyping. So looking at EPCAM expression versus CD103. With GM-CSF, you get upregulation of CD103, this is known. Maybe a little bit of EPCAM, very little. When we add just TGF-beta by itself, we get a huge increase in TGF-beta in those cultured cells. When we add both, we get almost all of them turning on EPCAM. That's just quantified here. I'll show you a bit more evidence for this in a moment. But this is the model we're actively working on, is that we believe what's happening is that pre-dendritic cells likely are entering the lung, responding to FLT3 ligand and differentiating into this early state, this vascular associated or perivascular CDC1. And then the lung is very rich with factors like GM-CSF and TGF-beta, particularly around the airways. So as they get recruited to that site for reasons we don't yet understand, we believe they're responding to those cytokines and further evolving into these airway surveilling populations. So all well and good in vitro, but we see something very similar in vivo. So we utilize the CLEC9 CRE system crossed to TGF-beta R2 flox mice. This is just showing the level of CDC1 specificity we have. So it's very efficient at CDC1s. We do get some labeling of CDC2s, so keep that in mind. But so we can basically knock out TGF-beta signaling in CDC1s. When we do that and look in vivo, phenotypically, we lose that EPCAM expression. So when we have CRE positive mice that are TGF-beta R2 flox, we lose responsiveness to TGF-beta, we lose EPCAM expression. So certainly TGF-beta is driving EPCAM expression at some level in vivo. When we do our IV-IT labeling assay, we dramatically impact the airway surveillance function of these cells, or at least the airway labeling of these cells. In the absence of TGF-beta signaling in those CDC1s, we get very inefficient labeling in vivo. Interestingly enough, we don't get a huge impact on the IV labeled populations. There's roughly the same. That kind of makes sense. We're shutting down one half, but it doesn't necessarily seem to be like an increased recruitment of the IV labeled in the process, or like a retention in that compartment. Something to be understood. So the last thing I will leave you with, very much we're now going into the territory of, hey, we think this matters. It all is consistent with something that matters, but there's a lot that could be going on here. But when we go back now to our metastatic assays and say, okay, we've predicted that this vascular surveillance network exists, the TGF-beta is likely driving cells to the airways or retaining cells at the airways, we're not sure yet. in the absence of that pathway, do we then have better vascular surveillance and reduce metastatic burden? Short answer is we do get reduced metastatic burden. The answer to that first question, we don't know yet. I think I showed you already that the IV labeled cells are relatively consistent. So it's not like we're likely getting a huge enhancement of vascular surveillance. We do actually see a slight increase in loading in the IV labeled cells, but I'm not quite confident about that yet. But this is also an immunosuppressive cytokine. We could also be altering the immune environment, and we need to dig a lot deeper on understanding that. But this is kind of how we're starting to think about this. I'm not showing you any viral data today, but we are doing experiments looking at influenza, and we have some pretty compelling data that I hope to share in the future on the airway surveillance side of this equation. But we think that there's a developmental trajectory within lung CDC1s that has functional consequences at both the vasculature and the airways. We believe GM-CSF is part of this equation. We believe TGF-beta is part of this equation. Basically, engaging with DC differentiation and driving this, driving this airway surveillance phenotype. And conversely, the early cells in this process we think are likely important for early surveillance against metastasis, though perhaps inefficiently so. If we impact this TGF-beta signaling pathway in CDC1s, we compromise that airway surveillance phenotype, and we at least putatively are enhancing anti-metastatic immunity, though we don't know all the underlying features of that yet. So I will end there. Happy to take any questions. Just to acknowledge folks at the Fred Hutch and other collaborators, our collaborators on Infinity Flow and my old lab, funding, and then my lab here. Much of the work you saw was done by a former postdoc, Shannon Ludall, and a really talented postbac who's now in grad school at University of Colorado, Juliana. And then currently, Sajida and Victor are working on this project, and Radhika, postdoc, and Emily Park support them and have their own projects that I didn't really talk about today. So I'll end there. Thank you.
I've got microphones.
Hi, is the TGF-beta signal just a priming event, or do they need constant exposure? And then where does that exposure come from? Is it the endothelial cell, or is it once it enters like the parenchyma?
So the very short answer is we don't know yet. So we've modeled that in vitro. If we take away TGF-beta as efficiently as we kind of can in vitro, just like washing the cells, they maintain EpCAM expression. So it's not clear in that context if it's required for maintenance. We don't have a CRE-ER system going yet for that, though it does exist to maybe address, hey, if we take it away midstream. So I don't know for sure. As far as the source, that's a very, very interesting question. That now that we kind of have a sense that this pathway probably is doing something, I think it's a very worthwhile question to ask. It's unlikely the TGF-beta is coming from the endothelium based on analysis we've done of like single cell RNA-seq. It's very highly produced by lots of cells, but in particular airway epithelium and like fibroblasts around the airways, things of that nature. So that's my prediction. There are tools that one can get at. There's TGF-beta flox mice that you can cross to like SBC-CRE, for example. So we're planning those, but we haven't gone down that path yet.
So you're describing these IV labeled DC as ones that are sampling in the blood, but then you also showed that newcomers that are going into the lung from the blood would be labeled as well.
Yeah.
Any kind of sense of what sort of proportion you're picking up there of stable lung labeled DC versus ones that just arrived this morning?
Yeah, that's a good question. So the best answer I have to that is from our depletion experiment. So it seems like that process of entry takes a couple of days.
That might depend on the decay rate of DT.
Yeah, that's fair. That's totally fair. So I guess, honestly, an answer, I don't know for sure. The one thing I can say is when we look in circulation, like just sampling the blood, you don't really ever see XCR1 positive, MAC class 2 positive, CD11C positive cells, unless you, like there's some autoimmune models that do develop that, but steady state, we just don't see it. We do see pre-DCs. We see the CD11C positive, class 2 negative cells, CLEC9 positive, if you look at a reporter. So within the pool that we're showing as IV labeled, those are 100% XCR1 positive, class 2 positive, CD11C positive. So whether they're recently that or they've been that for a couple of days, we don't know. I think the best way to do that, maybe we could get clever with some like pulse chase experiments, but we haven't done that yet.
Okay, are there more questions in the room? Okay, hang on.
Thanks, that was beautiful. I'm wondering if you have any information about where the cancer is entering through the vasculature. Is it preferentially at the sites of where DCs are sort of splitting the vasculature open and then giving the DCs easy access to them, or are they sort of avoiding it, or is that a factor in metastasis?
Yeah, that's a super good question. I don't think I don't think they're predominantly entering where the DCs are. And that's based on quite a lot of imaging we've done trying to find DC tumor interactions. The vast majority of tumor cells that end up in the tissue are not proximal to a DC. But that's actually kind of interesting. It actually fits our model slightly better that perhaps where DCs are localized, that's where you get microlocalization of NK cell activation and better clearance in those sites. That's a stretch, right? But that's how it would shake out if things are working the way we think they are. In contrast to that, we do see very clear evidence of monocyte macrophage populations around cells that have entered the tissue. So if we look at, say, 16 to 24 hours at the tumor cells that have survived past this kind of early checkpoint, they almost always have like a nice little niche of macrophages. So that's what we think is probably mediating that effect, if not the tumor cells just kind of having some intrinsic MMP9 expression, for example. And that's quite consistent with work from like John Condelis that has shown on the other side of the equation that there's this thing he calls TMEM that is like a tumor macrophage endothelial kind of network that is important for entrance into the vessels. We suspect there's probably something similar going on, but it's not really cleanly worked out. And also, sorry, it's very quickly, in the context of our model, IV model versus when we do, say, have a subcutaneous tumor or a true spontaneous model, the vasculature is very different in those two environments as well. The primary tumor has a pretty significant impact on vascular leak and vascular permeability. So it might actually be different depending on the model we're looking at too.
All right, let's finish with an online question from Rick Jansen. In the RNA-seq heat maps where you look at expression patterns across 5 different DCs locating to the airway, blood vessel, and unlabeled, why do you think there is quite a bit of variation across most genes, even within each of the 3 groups? What about local environments do you think is creating the variation in expression?
Just want to make sure I understand which data. So if we're talking about the bulk transcriptomic data, we actually don't see a ton of diversity there. There's a little bit. Within the single cell, there's quite a lot. And so I take care not to overinterpret single cell RNA sequencing data. There's a lot of dropout. So I might be missing the point of your question. I apologize. Please feel free to reach out to me if so. But in like, say, the trajectory plots I showed where we're looking at expression that way, much of that heterogeneity is due to detection versus dropout, and I don't think it's necessarily intrinsic heterogeneity. Other than that, in the bulk RNA-seq, it's actually pretty consistent within each population. Across populations, I think it's driven by the environment. Like the airway cells are seeing a different environment than the vascular labeled cells. So hopefully that answers it. If not, please feel free to email me or.
All right, let's say thank you one more time to Dr. Headley.